Good morning. Welcome to the second day of this presentation of the workshop. And today we're going to be talking about data analysis. So the first part of this presentation, I'll just go over generally what we do for data analysis, how we take raw data that comes out of the system, process it such that Allele Calling software can read it in and assign the proper alleles to each of the locus. Secondly, in terms of the data analysis, we'll go over the data that we generated yesterday on the demonstration runs, the two runs, the one that has a slow or the standard protocol, and then the one that had the fast protocol. Go through each of the lanes, and then after that, that would conclude data analysis. But feel free through this part to ask all the questions, make all your comments, because this can be a very interactive part of the presentation, especially when we look at data or when we're going through the data processing part and talking about how data goes from raw data over to processed data. So in terms of data analysis, if you look at a trace of raw data, it typically looks like this. It's really messy. And by messy, I mean, first of all, what you see are baselines that are not all lined up together. Secondly, you see bleed through. For example, here, this would be a blue dye that's going through the system, but then the blue detector picks it up, the green detector picks it up, the red detector picks it up as well. So for one dye, you get signals from all your detectors. And then also, you'll see that the baseline is rough, so it has more perturbations that you normally see out of a fully processed trace that you would typically get. So the first thing that happens with data processing is smoothing. So what smoothing does is it essentially takes this portion of the baseline, which is the noisy portion, and puts a smoothing filter onto it so that it becomes much smoother. You don't get all the little noisy spikes. The smoothing filter that is typically used on many software packages is not just an averaging device, but rather it has a specific function and form to it such that you can smooth out the data but not affect the width of the peaks. So if you look at the peaks from the raw data, which is this one here, you'll see that the peak width and the peak shape is essentially the same. The main difference between the two traces after smoothing is that the baseline, which is this portion or this portion, has been smoothed out. So that's an important function for of software, and that is to take the raw data initially and smooth it without really either changing the height of the peaks, the width of the peaks. And after smoothing, some software has spike removal capability, and that is for peaks that are very narrow, generated by electrical perturbations. The software will just go through and figure it out and just eliminate them. After spike removal, usually what happens is color correction, and this is usually one of the largest parts of data processing software, and that is how do you go from a raw trace which for a given dye, for example blue, has signals in both the other colors, green and red, to one where you would get only blue but nothing on the red and nothing on the green traces in this case. And that's a function of color correction. And we talked a little bit about it yesterday, but color correction comes because of this effect here. So if you take the example of 4 dyes, which is the spectra I show up here, you have 5-FAM, which is blue, you have Joe, Ned, and Rox. First of all, notice that the visible area for detection is somewhere between 520 nanometers, which is here, from this point up until 650 nanometers of wavelength. So the window is relatively small. Your laser excitation is somewhere between 488 nanometers and 514. So you'll have peaks that are here that are really associated with your laser. So for detection, you want to make sure that you only detect within the visible window of around 520 to 650. If you look at the spectra for each of the dyes, so take for example 5-FAM, this one here, you see that it's actually relatively broad. So even with the dye that is at the closest end to the lower wavelength side of the spectrum, it's broad and it has a tail that runs all the way up to 625 nanometers. So if you have a detector, in any of the visible portions of the visible spectrum that you want to detect, 5-FAM will make a contribution to it. The same goes for the green, which is Joe, Ned, and Rox. Typically, the width of an emission peak or a dye is about 30 to 40 nanometers, and that's this width over here. So as you can see, it's really difficult to pack in more than about 4 or 5 dyes and have enough separation between them so that you can adequately detect them without any confusion in colors. The second thing about detection is, as you remember from yesterday, all the wavelengths come into the detection box. And from this detection box, a number of mirrors and bandpass filters are used to split this wavelength into 5 different detectors. So what I've represented here is the detection window of a detector. In this case, it's around 20 nanometers wide, and that would be the bandpass filter. And it's centered, for this example, on the blue. So this would be detector number 1, designed to detect blue. Detector number 2 would be designed with a bandpass of wavelength to detect green. Detector number 3 would be for yellow, and detector number 4 would be designed for red. And for this specific example, GeneBench has 5 detectors, so it also can detect the orange, which is the lizamine signal that's out here by the 650 nanometer wavelength. So if you can see, if you were detecting, for example, looking at the green detector, or the one that's optimized for the green dye, you'll see that when a green dye comes in to the detection window, it'll pick up a good, strong green signal. And that's really what you want. But at the same time, detector number 1, which has a window in around the 520 to 540 nanometer range, will also pick up this portion of the green dye. And the same goes for detector number 3. It'll pick up this portion of the green dye. And then detector number 4 will pick up this portion of the green dye. So at the end, for a given green dye that goes through the detection box, you'll get the strongest signal coming out of detector 2, which is your green signal. But then detector 1 will pick up some of it, a small fraction. Detector 3, which is really close, will pick up a larger fraction. And then detector 4 will pick up also a fraction of this green dye. So if you wanted to sort of take a rough guess, for detector number 2, you may get, if you had 100% 1 for signal, detector number 3, you would get 0.7 or 60%, 30% for detector number 4, and that's the area of this portion over the green. And detector number 1, you may get 0.2, a fraction of 0.2 of the green signal. And this is really what color bleed-through is all about. So we talked about the green signal. So for the green signal that comes through, all the other detectors will pick it up. For the red signal that comes through, or red dye that comes through, you'll get pickup on the yellow detector and maybe a little bit on the green detector. So every detector will essentially detect a portion of the dye that's coming through. So how do you deal with that? The whole objective of color correction is essentially taking what you call detector space, so the signals that you get from your detectors, and converting it into dye space. Because essentially what you want to know is, if I have one green dye coming in, I detect it with my 4 or 5 detectors, I want to know how many, at the end of the day, how many green dyes I actually have. So physically, from the DNA, one green dye will be emitting. Your detectors will have a mix of signals from detectors 1, 2, 3, 4, and 5. But then you want to somehow take all these 5 signals and say, if I have these 5 signals, I know that I have 1 green dye. So you're transforming from this detector space of detection signals back to this dye space, because at the end of the day you want to know how many green dyes I have, not whether my detector 1 picks up half of a green dye, detector 2 picks up more, and so on and so forth. So what it comes down to is really just linear algebra to take care of this. And we'll go through an example because I like this stuff. We'll go through an example, but it actually comes quite, at least I hope it makes it quite clear at the end, because this color correction always seems to be this magical thing that happens, and then out comes your result. So before I go to that, first saying that you go from sort of a set of detector signals, and you want to convert it, all these detector signals, into the dye space, or the number of dyes you have. So if you have sort of this type of function that you need to use, then linear algebra is the way to go. And if you want to do a lot of linear algebraic manipulations, then you can use matrices. But since it's, I'm just going to give you one example. I'll just use linear algebra because it'll make things quite clear. I'll first start off by going back to this plot, and we'll use the example where you have a number of green dyes coming through, and on the detector side you'll have 5 detectors. So detector number 1, or 4 detectors in this case, detector number 1, 2, 3, and 4. Detector number 1 will say, when this green dye comes through, I'm going to see 1,000 counts on my detector. So we'll just put 1,000 here. Detector number 3, which is adjacent to it, a longer wavelength detector, we'll see a smaller portion of this green signal, and it may report back 600 counts. Detector number 4 is going to be way off, so it's going to see less, this small portion. Detector number 4 will see only this small portion over here, so it may see 200 counts. And then detector number 1, which is only seeing this portion of the green dye, may only see 100 counts. So what you get back from data from your signal is, for a given count, you'll have detector number 1, 2, 3, and 4. 1 reporting 100 counts, 2 reporting 1,000 counts, 3 reporting 600 counts, and then 4 reporting 200 counts. What you really want at the end of this is that, and this is called detector space. What you really want is sort of the dye space, and that is when you make the transformation from detector to dye, you want it to say detector 2, which in dye space is green, will have 1,000 counts. Detector 1, which is in dye space is blue, will have 0 counts. Detector 3, which is yellow, should have 0 counts, and detector 4, which is red, will have 0 counts. And from a graphical perspective, what this looks like is the color-corrected trace that you get on the instruments when you start off with your raw data and you see, oh, 1 red peak or 1 green peak with no pull-up associated with it. So how do we get from detector space to dye space? Well, you start off by saying, I have detector 1, which is 100 counts, detector 2, which is 1,000 counts, detector 3, which is 600 counts, and detector 4, which is 200 counts. I want to make some type of transformation so that I end up with only 1,000 counts on detector 2. So it looks pretty simple, because what I have to do is I have to subtract 100 out of detector 1, 600 out of detector 3, and 200 out of detector 4, and I'll only be left with 1,000 on detector 1. And that's fundamentally what's happening with color correction. You generate a matrix, and the matrix is actually this linear algebra manipulation of it. So you always see this matrix 4 by 4 and 5 by 5. What that really means is that you're developing the coefficients so that you can go from detector space to die space. And I'll just be a little bit more specific here because you may be saying, okay, this is kind of weird because it just seems too simple. Or, you know, I'll just take 100 off here, 600 off here, and 200 off here, and you're there. And it actually is that simple. But first of all, before we get to that point, usually what happens is for every signal that comes through, they're not all the same height. So if you have one nanogram of template that you run through the system, you may get 1,000 counts on detector 2, but if you ran 0.25 nanograms, you'd get a quarter of that, you'd get 250 counts. So it's not as simple as just a straight, I'm going to delete 100 out of every signal that I receive. So what people normally do is they first normalize the detector counts for a given die, and that's what they call the color ratio matrix. So at the end of the day, what you get is for your color ratio matrix that you generate, you normalize it and you take the inverse of it. And this will let you get, for your green signals, all of the detectors that it picks up, it subtracts the ratio to get you into a space where in the die space it just reports only green but not anything in blue, in yellow, and in red. I don't know if I've lost anyone or everyone, but if you have questions, we can get into a little bit more detail, or if you want, we can just go through the whole thing in detail. But that's generally what, that's sort of specifically what color correction involves, just taking the ratios of the signals from each of the detectors relative to the one you know. So for example, if it's a green dye that comes through, you want to eliminate the contribution from the blue dye, you want to eliminate the contribution from the yellow dye, and you want to eliminate the contribution from the red dye.
Is your algorithm doing this for each peak within each sample, or is it doing it for all green peaks, for example, within a particular sample, or?
It's for a given. So that's a good question. So for a specific run, all of the green peaks, green dyes that are detected will have the same spectral contribution as read by all the detectors. So what we do is for a given run and for a given lane, we apply the same color ratio matrix to all the peaks. And so all the peaks that are green. And then we do the same for all the peaks that are red and all the peaks that are blue and all the peaks that are yellow as well. So we can go from, just like in this trace here, where you have, for example, this is a blue dye, where you have blue, red, and green. It would be corrected into just a blue dye with no contribution from red or green. The same color ratio matrix, or the color correction matrix, which is the inverse of the color ratio matrix, is applied also to this blue dye, and it gets you this result as well. For the red dyes, it has its own color ratio, color ratios that are used. So but all the red dyes will get converted to the red with the same transformation. So either I'll have a question or we'll, yeah.
The way the detection system works, stop me if I go off on this. You've got your scanning mirror that looks at each of the 16 capillaries, channels, what, 5 times a second? Is there ever any problem created by the disparity between the amount of time it takes to scan over the 4 colors or 5 colors in each of the 16 capillaries? Do you get a difference because the peak, the signal is stronger at the moment that it's scanning the orange channel and the moment it's scanning the green channel?
Oh, yeah. So when it's scanning one channel at a time. But it detects all of the wavelengths at the same time. So there's no disparity in the wavelength. There is effectively a time difference in the scan, but ever so small between lane 1 and lane 2 and lane 3 because it's moving across.
all the detectors detect all the colors simultaneously.
That's correct.
For each channel. Okay, I got
That's
it.
correct. Yeah, so that's the basis for color correction. I hope I gave you some sense of what actually happens from going from signal into a detector into dye space. And so the next part of this presentation is I'm going to switch the computer over to this other mode here so that we can call up trace processing software. And the trace processing software that we use is commercially available. Our system is not doesn't have to run with this particular trace processing software. It can run with essentially any software that is available in which we can transfer the data into. So the software we're currently using is GeneMarker HID, which is a commercially available piece of software. And what it does is that it can import data from our system. We've formatted our data so that it's compatible with it. And I'll show you what happens here. So it pulls it in. It can import data into the system. And this is the raw data that it imports. And then from then on, it can also do the allele calling processes. So what you would do is select a panel, tell it what size standard you use for the specific kit that you're using, what color the size standard is in, and then move forward. Then it has some processing parameters, which includes the number of bases you want to call from and to, some peak detection threshold capability, and auto-ranging capability. So by going from that point, which is the raw data that's color corrected, it'll then place it into a format that can be used for allele calling, what you would normally use in your lab, I assume with GeneMarker or GeneMapper. So before I get there, I just wanted to show you what data looks like that's not color corrected through our trace processing software. So if we just imported our raw data into GeneMarker, what you would see is essentially this. This is the DS33 size matrix standard. And what you see here are the peaks, the orange, red, yellow, green, and blue peaks. And what you see is that for the blue peak, as you would expect, you get blue, green, yellow, and red trace contributions coming from each of the detectors. The same holds true for the green peak, and you have more green than yellow than red than blue. And as well, the yellow peak, so you have more yellow than red than green than blue, and then the red peak. And the orange peak, which is on the far side of the wavelength window, sees both only orange and red. It sees very little yellow, green, and blue, and we expect that because it's on the other side of the wavelength window. So I just wanted to show you that because the next thing I'm going to do is pull up a trace that has been corrected through software on GeneBench. So it takes the raw data now, it processes it so it's color corrected, and it moves it into GeneMarker. We can start with we can go with the fast run, for example. And the first thing we have to do with this software is really just to make sure that the ladder, the Allelic Ladder, is calibrated with the proper alleles. And what this involves is essentially just taking the Allelic Ladder sample, which we loaded in lane 6, and just running it through the software so that it knows that the specific Allelic Ladder is being used and the sizes for that Allelic Ladder. So I'll do that right now. This is the raw data from GeneBench, and what I'm going to do is ask this software to process it. Get back here. Scale it. And then we're going to call the Allelic Ladder. Since this is a fast run, the start range has to be changed to incorporate only the small piece of data that we use. I'm going to call only from 1,000 to 360 bases, 100 to 360 bases. And I'll calibrate by clicking this Create Virtual Ladder. So the ladder is created, and what you see from here is essentially the Allelic Ladder. The Allelic Ladder, all of the loci and the alleles that are called. If you want to zoom in on THO1, we can just check on the resolution. And you see that 9.3 and 10 has quite high resolution, as we would expect from the discussions yesterday. So the Allelic Ladder is complete, and now what I'm going to do is just load in the rest of the lanes. Since we already created the virtual ladder, or the Allelic Ladder, I'm going to click off this button. And then it'll just go through. And what it does is first call the sizes and then call the alleles. First of all, you see lane 16 has failed, and it's failed because that's the DS33 sizing standard, so matrix standard. So there wasn't any sizing standard associated with it, and couldn't find it, and it was unhappy. The first thing we do is, when we go after calling, is first just to check to see if all of the sizes of the sizing standards have been designated. There's a score of greater than 80 typically means that good size match has been accomplished. So if we go down this list, you'll see that for lane number 4, which is the best match, all of the GS 500 peaks have been called and identified. The same goes for the other lanes. And this is just a quick quality check, at least when we run the software, to make sure that the alleles will be called properly and is not miscalled as a function of the improper designation of the sizes by the software. You'll see we turned on the 250 and also the 340 peaks included in our sizing. So firstly, let's go to the Allelic Ladder. We can look at one color at a time. you'll see that all of the alleles have been designated from the allelic ladder. Nothing out of the ordinary. Same for green. Yellow. And the red. And the sizing standard. So that was the allelic ladder. We can look at the 1 nanogram samples, which were loaded in lanes 13, 14, and 15. And for the blue traces, you see that we're getting signal strengths approximately for these ones, the heterozygous ones, of around 1500 RFU. And this is 1 nanogram of input. This software doesn't have stutter elimination thresholding. So what you will sometimes see is stutter being called as well. And that's what this 12 and 12 allele is. So this is 1 nanogram. And in the green trays, also around 1500 RFUs on average. The yellow trace, which is a little bit noisier down here, appears to be called by the software. So these little bumps on the baseline, which is above the calling threshold, get identified. But they're clearly not peaks. This is the red trace. And then if we go down to the next set, which is the 0.5 nanogram, you'll see essentially the same thing, except the signal strengths are now half of the 1 nanogram. So these are approximately 700 or 800 RFUs. If we go down to the 0.25, you'll see essentially the same thing, less RFUs, and because the scale is now starting to get compressed, you'll see the baseline noise much more prominently. And then when we get down to the 0.1 nanogram, which are the first 4 samples, samples 1 to 4, we'll see that we have signal strengths in the approximately 100 RFU range for 0.1 nanograms. For blue, we get still very good signal to noise. We still get good signal to noise on green, but the signal strengths on green are slightly less now than the calling threshold, so you don't get full profiles, or specifically these alleles are not being called by the software because we set a threshold of 100. Yellow is starting to get noisier and the alleles are still present, but some of them are not called. Red, and then the sizing standard. So that's sort of the analysis from the fast run that we performed yesterday. And that's typical of what one would get out of GeneBench for STR separation detection and then analysis through the software package. Are there anything specific you want to look into before I move off this trace? No? Okay. So now we move on to the second run because we made 2 runs, so we'll analyze both of them. Save changes, no. First thing I do is load in the ladder. To change the start sizes, because this is the standard run. Create a virtual ladder. And just check that all of the alleles are called. All the alleles on the allelic ladder are called. The resolution on the fast run we took a look at earlier, and this is the resolution on the standard run. They're approximately the same in high. And then what I'm going to do now is just go to the folder and retrieve all the traces from the other lanes. Okay, so it's processed all the 16 lanes of data. Again, lane number 16 is the DS33 standard, so it's failed because it doesn't have a size standard associated with it. And we check on the sizing scores for all of the 16 lanes. You'll see that from lane 1 to lane 14, we get good sizing, but lane 15 has a low sizing score, 56. And you'll see that the software somehow did not pick up enough of the peaks. These two peaks, I think, are clear enough to identify as sizing peaks, but it didn't pick it up. So what I'm going to do for this specific lane is to, we can either add the peaks or just not analyze it and consider it a failed lane. For the other lanes, lane 1, or the sample number 1, all the sizing peaks have been identified. Sample 2, sample 3, 4, 5, 6, 7, 8. And 14. Okay, so what I'm going to do now is just go through the same format of looking at the 1 nanograms first. You'll see that for the standard run, the signal strengths are also around 1500 RFUs for the 1 nanogram sample. Again, the allele 12 is just the stutter peak from 13. move on now to the next set, which is the 0.5 nanogram. You'll see now they're approximately 800 RFU or 750, which is essentially half of the 1 nanogram template. All the alleles are being called. For the yellow trace, the alleles are called, and the software is picking up spurious, just noise spikes on the baseline.
You see that more in yellow than the other colors. Is there a reason behind that?
Yes, that's right. The yellow dye from the AB kit is weaker, so what ends up happening is that we detect less signal from it and more baseline. And then red. And this is the 0.25. And you'll see for red, for this specific trace, there's an extra peak that's called. So this comes into question: is it really a peak, or is it some type of color correction issue? And for this particular peak, we can check. So I can turn on all the colors, and what you'll see are these are the two red peaks that we are expecting to get. And this is the red peak that's spurious. And what you see here, though, is that there isn't another color that's overlapping or underlapping this trace. So this peak is actually real. Its source I don't know where from, but it's not a function of the color correction generating a spurious peak. And then finally we go to the 0.1 nanogram samples. And you're starting to see some alleles not being assigned because they fall below threshold. And the same for yellow. And the same for red. Yep, so in terms of data analysis, that's what I'm presenting. And if you have any questions, I'd be happy to go through any of these channels or lanes and look more closely.
Can you go back and show us the size standard on lane 15 again?
Lane 15.
This is the one where a couple of them didn't come up. Wasn't it? Or they didn't get assigned to the size standard?
Oh,
Yeah,
that was
two
lane
peaks.
10. That was lane 10. This one. Right, these two were not assigned.
Why? Is that just a size issue where the peaks aren't big enough to meet a threshold, or why would those
this
not be
part,
assigned?
I'm actually not sure why. Mainly because this part of the software we purchase, and it's some algorithm that goes ahead and assigns and identifies peaks. You know, just by eye, these two peaks don't look any less different than, say, this peak or this peak.
The ones that are assigned they're the correct values.
Right. So it must have
Missing
some
peaks.
thresholding or some rule that it's using that unassigns these.
How do you use DS33 in this I mean, I know it didn't size, but then what would be the point of loading it if you're not going to look at
Oh, so I would. There are two cases. One is if you were using it in a lab or using it in an analytical application, you would load DS33. DS33 is just something we load in our lab as a check to make sure that the color correction is always functioning. So I can show you how we use it. What we do is we look at the raw data, which is, oh, I have to restart this. We actually just look at the raw data in this case. So it's actually thinking. Okay, so the DS33 is loaded in lane 16. So what we do in our lab is when we call up the software, the first thing we go to is the DS33 size standard, and we check on it and just take a look to see if blue is truly blue. In this case it is. No residuals from the other colors, green, yellow, and red, and orange. So this just gives us confidence that, again, the software trace processing has functioned, and if there are any issues related to software, we know where these issues lie. Either it's on our end, and that's the trace processing before we put it into the software, or it's somewhere else. It's more an engineering tool.
Is there like a size limit or something that you look at, or like what would a bad one?
Oh, a bad one. So for example, I can let me add so let me remove all the samples and add only lane 16. This is going to be really, this will show what the differences between the good and the bad one are. So I'll add lane 16. And essentially what I've done is I've added two versions of the lane 16 that we have. The version up here has been processed by the trace processing software. This version down here is raw data, which has not been processed. So you'll see if we just zoom in on the blue, the raw data has overlap. So this is only the blue trace, but then you see green, yellow, and red as well. But after trace processing, you only see blue. So somewhere in between, this is the worst case you would get. If you had poor color correction, you may get a little bit of another color, say for example, red or green, and it would be relatively small, but not in the level of the baseline. And what this would end up happening is it would generate usually a peak in your allele, and it would be designated as an allele because it'll most likely be higher than the calling threshold if it wasn't corrected properly.
When would you tend to see that? Say you're using this as an engineering tool. Is it when a chip's going bad, or?
When would you tend to see that? You see that we have an algorithm that calculates the proper matrix to use from DS33. You would see that if this algorithm failed. And this algorithm, instead of getting the proper ratios where it would be 1.6, for example, 0.3 and 0.2, it may say 1, 0.65, 0.3 and 0.2. And in other words, you know, for the main peak, you would still get the right signal, but somehow the algorithm made a miscalculation, assigned too little subtraction on the next color, and you would end up with that peak. We typically don't see that. We saw it in the early days when we were starting to write this algorithm, but we typically don't see this anymore. where it would miscalculate, because we've been refining the algorithm for calculations.
When you were going through and processing the data initially, you were pressing all sorts of buttons up the top. Could you explain what you were doing?
Yeah, so each piece of software is different, and each piece of allele calling software is different, and and they all have functions that can be used. So in this software, there's spike removal, there's saturation control, there's smoothing, there's baseline subtraction, there's auto pull-up removal, and there's manual pull-up correction. The trace processing software that I've implemented for GeneBench, I've just implemented only the color correction part of it. So color correction and baseline subtraction. So it doesn't use any smoothing. It doesn't remove any spikes. And it also doesn't scale the data so that a 1 nanogram template has approximately 1,500 RFUs. So what I had to do was, because I didn't implement all of those functions in the initial trace processing software, I just implemented the built-in functions here. So what I did when you saw me clicking was spike remove. I clicked some smoothing, and then I manually corrected, or in other words, just scaled the data so that we got 1,500 RFUs for a 1 nanogram sample. And those were the buttons I was pressing: spike removal, smoothing, and manual pull-up correction. For the case where one is using the software in a sort of an automated fashion, I believe this package has macros that one can implement. So for example, in this portion, I could have clicked on smoothing, peaks, saturation, so on and so forth.